Our lecture today is the second part of muscle tissue concerning the cardiac and smooth muscle. Cardiac muscle, they are striated but differ in skeletal muscle in certain points. They are bifurcated rather than in form of cylindrical tubules with an anastomosing network. Each myocyte have one or two central nuclei, unlike skeletal muscle which have multinucleated peripheral located myonuclei, and more sarcoplasm. They are more larger and developed mitochondria comprising 40% of the cytoplasmic volume and containing mainly type 1 fiber, which are high oxidative slow twitching fibers, containing certain structure called the intercalated disc that found at the site of junctional complex between the adjacent myocytes and undergo rhythmic, spontaneous and involuntary contraction. The glycogen is present in skeletal muscle, in cardiac muscle, and absent in other type of muscle tissue. Lipofuscin granules are present within the myocyte. These are granules that represent residual structure within the sarcoplasm, and it is found in different organs and indicating aging process. So in this figure we can see the cardiac myocytes. They are bifurcated, not straight, and we have this junctional area in dark color called the intercalated disc, and the nuclei are centrally located. You can see the striation is present in the cardiac muscle. In this figure also we can notice the striation and bifurcation of cardiac myocyte, and the arrows pointed toward the intercalated disc, which is a junctional complex. Intercalated disc represents a junctional complex found between adjacent myocytes. It contains a straight line or a form of step-like junction. The step-like junction containing two regions, the transverse region which is rich in macular adherence, which is desmosome, and zonular adherence, which is important for providing a good cell adhesion, while the lateral region containing gap junction that is important for spreading of excitation between adjacent cells. In this figure we can see the cardiac myocyte, and here we have the transverse region rich in macular adherence and zonular adherence, while the lateral region, which is parallel to the myofilaments, rich in the gap junction. The sarcoplasmic reticulum and transverse tubules are found in the cardiac muscle, but with certain differences. The tubules, which are invagination from the sarcolemma, are more numerous and larger in the ventricular region than in skeletal muscle, and they are found at the region of Z band, while in the skeletal muscle they are found at the region of AI junctions between the bands. The sarcoplasmic reticulum is not well developed, and it is found irregularly between the myofilaments. And here we have a diet, which is one T-tubule with one terminal cisternae of sarcoplasmic reticulum. Unlike the skeletal muscle, we have a triad, which is a single T-tubule with adjacent two swelling of terminal cisternae of sarcoplasmic reticulum. The myofilament arrangements also differ, and we have the atrial muscle smaller than the ventricular with little T-tubules, and we have specific granules in the atria called atrial natriuretic factor, which is responsible, found in the wall of the atrium, and it is concerning certain types of arrhythmia. In this figure we can see electron microscopic figure for the cardiac myocytes. At the atria we have the nucleus, and we have these granules, which is the site of natriuretic factor, which is a polypeptide found in the sarcoplasm. The conducting system of the heart made by the impulse generating system, which is the SA node, or called the pacemaker of the heart, from which waves of activity pass towards other part of the conducting system comprising the AV node, atrioventricular node, found at the AV junction, while the SA node found in the wall of the right atrium. Then we have the bundle of His. From the bundle of His we have bifurcation of the bundle branch into right and left side for ending toward the Purkinje fibers. In this figure we can see this is the heart, so this is the right atrium, in which we have the pacemaker of the heart, which is the SA node, then from which we have the atrioventricular node. In this region from the SA node to AV ventricular node represent the PR interval of the ECG. Here we have the atrioventricular node, we have the compromising the QRS complex, then we have the bundle of His, and then Purkinje ending in the Purkinje fibers. Here we have the left bundle branch and we have the right bundle branch, sometimes blocking to some region of the bundle branch, leading to bundle branch block and leading to specific types of arrhythmias. Purkinje fibers, which is the termination of the conducting system of the heart, they are found in the subendocardial region, and here we have the endocardium. In the subendocardial region we can see the Purkinje fiber. In higher magnification they are larger than muscle fiber or cardiac myocyte, containing a central nucleus surrounded by perinuclear clear area due to the presence of glycogen. Have a well-developed sarcoplasmic reticulum with little contractile myofilament and lacking the T-tubule. The other part of the muscle tissue is the smooth muscle. They are involuntary, non-striated, undergo hypertrophy and hyperplasia, and unlike the skeletal muscle undergo hypertrophy only. Also cardiac muscle undergo hypertrophy, no hyperplasia in the skeletal and cardiac muscle. Smooth muscle capable for regeneration. They are originated either from mesodermal origin or through mesenchymal cells, and sometimes we have a type called myoepithelial cells containing smooth muscle filaments are originated from ectodermal origin. They are found in different locations in the internal organs, such as GIT, respiratory tract, wall of the blood vessels. In the skin is in a rectal pili muscle, in the eye, the iris, diaphragm, uterus, and the myoepithelial cells found in the glands, duct, and the acini. Other regions also can be found. They are spindle in shape. They are tapered toward the end, with central larger region contains central single nucleus with multiple nucleoli arranged in form of layers, sheet, or enclosed by basal lamina. They reaching to up to 50-100 micrometer in length. They are less in sarcoplasmic reticulum, no T-tubules, and the myofilament is not regular arrangedly, such as in sarcomere of skeletal muscle or in cardiac muscle. They are found in form of lattice-like pattern. In this figure we can see longitudinal section of a smooth muscle. We can see the spindle-shaped nuclei, and this is the cross-section, which is called mosaic pattern of smooth muscle. Also in this figure we can see the spindle shape of this cell, and we have the central nucleus. In the cross-section we can see either without nucleus and with nucleus, according to the side of the section from the smooth muscle cell. Contraction of a smooth muscle, it occurs through contraction of lattice-like myofilament, and here we have a dense body in a state of Z disc. This relaxed form, and this is a contracted form. So we have a quick review. This is a skeletal muscle, multinucleated, striated. This is a cardiac muscle, branched, and we have the intercalated disc, junctional complex, and we have the smooth muscle, spindle in shape with centrally located nucleus. Regarding regeneration of muscle, the skeletal muscle, we have a group of cells called satellite cells responsible for their regeneration, while cardiac muscle only regenerate in children. In adults forming scar tissue. Smooth muscle is capable for regeneration. And thank you for your listening.